In this presentation, we'll be discussing the importance of chloride ions in the central nervous system, skeletal muscle, and smooth muscle. You might be familiar with the movement of sodium and potassium across the plasma membrane, but what you may not know is that chloride has an impact on membrane potential and action potentials as well. Exploration into gamma-aminobutyric acid and its effect on neuronal excitability will be explained in this presentation. In addition, we will discuss a central nervous system depressant, Valium, and why its combination with alcohol and certain drugs is harmful. Finally, we'll explore chloride channels in skeletal muscles and discuss channelopathies among chloride channels. First, let's talk about a neurotransmitter that plays an important role in chloride ions, gamma-aminobutyric acid, or GABA for short. GABA is a primary inhibitory neurotransmitter, meaning that the neurotransmitter interacts with the receptors of a neuron to decrease the likelihood of that neuron to produce an action potential. The activation of GABA also causes the action potential to become more negative, which you may know as inhibitory postsynaptic potential, or IPSP for short. There are two types of receptors that GABA binds to, GABA A receptors and GABA B receptors. Let's focus on GABA A or GABA alpha receptors a little more. When GABA interacts with a GABA A receptor, it opens an ion channel permeable to chloride ions. The extracellular concentration of chloride is typically higher than the intracellular concentration. So, as chloride ions move into the neuron, this causes the membrane potential to hyperpolarize, which again makes it less likely for an action potential to occur. With the focus on GABA receptors, we are now going to explore central nervous system depressants. One central nervous system depressant that acts on GABA receptors is Valium. Valium is a benzodiazepine that is usually prescribed to patients suffering from anxiety disorders. When this drug is taken by itself, it is relatively safe, but the harmful dangers increase dramatically from combining Valium with other drugs and alcohol. This is because these substances also act on the GABA receptors. Substances such as drugs and alcohol have depressant properties, which suppress and slow the brain's activities and physiological processes. When mixing these with Valium, it affects the bioavailability of the other substance. This can cause overpoisoning and intoxication for not only Valium, but for alcohol and drugs as well. Additionally, this intensifies the effects of Valium's properties, causing the consumer to pose a higher risk of central nervous system depression. You may not know, but chloride ions play an important role in skeletal muscle movements. Let's explore this further in depth. The type of channel responsible for chloride movement in the skeletal muscle is the CIC1 channel. This channel is voltage gated with contraction and relaxation of the skeletal muscle, controlling the flow of ions in and out of muscle cells. CIC1 channels are made from two protein subunits, which are produced from a gene called CLCN1. These two proteins work together to regulate chloride ion movement between muscle cells. This same gene, CLCN1, can become mutated, and that leads to channelopathies on ion channels. Channelopathies are heterogeneous disorders resulting from the dysfunction of ion channels in the membranes of cells. The most common skeletal muscle channelopathy is myotonia congenita. This causes by the mutation of the CLCN1 gene. Muscles usually require a high chloride current for faster repolarization and stabilization of electrical excitability in the muscle. However, myotonia congenita causes a reduction in the chloride current, which results in the continuous depolarization of muscle cells. Alright, so thank you for listening to this presentation. I hope you gained some knowledge and insight into the importance of chloride ions in smooth and skeletal muscles, as well as the central nervous system. Thank you.